I also welcome you all, and it was mentioned, a warm welcome. I was one of the people arriving this morning at 4 a.m. And yes, I experienced the warmth. And I have to tell you, I like the warmth. And I'm not only talking about the weather. It's also the warmth, or it's not the right expression maybe, it's not my native language, but I know there are many warm people here, they are friendly, they are helpful. We've experienced our collaboration for 3 years now. The starting, I will show you, started 5 years ago, and it was a pleasure, and it's, I think, starting to become very successful. So I'm gladful for also the people hosting us here. Nias Ahmed, to mention, very important, the anchor here. People have already alluded on this acronym. I will explain that in a minute, what BRIDGE really stands for, but Nias is really the anchor, the pillar here on this side, and we are in Berlin. And then, of course, the bridge people have to walk across, and that should be the students, I think, both directions. Okay. So this is the official name, Functional Molecular Infection Epidemiology, complicated. I try to explain a little bit. I won't go into too much detail of science. There will be some later on. Hello, Mr. Fischer. And again, these are all the participating members, Freie University, and yes, I'm from the Charité, the medical faculty of Freie and Humboldt University. The International Research Training School, complicated name. And the DFG has the form for this for 4.5 years, strange number. First, students, they get a grant for 3 years, then the next students, and they are applying right now. An assessment center will select them, and then we go for another 1.5 years. And then the whole thing can be extended for another 4.5 years. So if you guys are all interested, for the next 4.5 years, we will have openings again. The speaker, of course, that I'm excusing today because he's moving his whole institute exactly these days. He wanted to be here, but he couldn't do it. To make sure the move goes well, you have to be there, and that's why I'm here as the vice speaker. Many other PIs. We have the Robert Koch Institute, which is in Germany. It's a little bit like the NIH in the States. It does epidemiology, very important institution. We have the Max Planck Institute for Infection Biology. And again, we have both the Humboldt University and Free University and Charité involved. And I mentioned Nias already, and there are many other institutes here. In my project, that I will mention a little bit in the end, the Mahavi Hospital is very important for patient recruitment and a lot of interaction. Students are at our lab right now and are applying for the next round, so there's a lot of interaction. Yes, we have, in Germany, maybe it needs to be explained. For graduate students in the lab, you do not automatically have funding, but the funding has to be applied for. A PhD student gets some money. Cost of living is higher in Germany. And these grants are provided for by the DFG, plus some consumables. And we have 2 postdocs that do central jobs in this whole network. So the second group is starting in October. The application deadline is over. I think the last I heard was that we received over 200 applications for these 11 positions. And we will select and then have an assessment center where everybody will present and then select the new students. Yes, there we go, 200. And of course then, and I made this little gap, we are working now already on the next round, the extension of the project for another 4.5 years. We have still 1 year to submit, but still we have to start. Okay. And these are the names. You may not know too many of them. Freie University, Susanne Hartmann just joined from the Humboldt. We used to have 2 Humboldt people anyway. And the Charité, it's a lot of people, but a lot of them have double appointments to projects. These in brackets, they have left. Usually they received a call to a professorship, so this is quite prestigious. For us, it means we have to replace them. I don't know if you can read it all. Robert Koch Institute, Max Planck. And now it gets even smaller, and I would say, it's warm here, the light is not that good. You can ask me about all the details. Those are the projects. I would just maybe summarize. A lot of projects deal with E. coli. It's a gram-negative pathogen, or not a pathogen. It can be a commensal of the gut, but sometimes may turn into something dangerous. In Germany, we had a big an outbreak with EHEC, dangerous bacteria, and you could track them down where they came from. And of course, NIAS works a lot on that, and Prosavila, and there's a big exchange. Then another focus is on Mycobacterium tuberculosis. I will focus on that in the end of my talk also, and there's of course interaction here. In Germany, we don't have too many TB cases, but here, of course, there are some, and so that makes a good interaction. So to study patients, it can be done much better here, and it can be done best in interaction. So these are the researchers, and then, oh, it doesn't work. Then there's also Helicobacter pylori with the world expert Thomas Meyer of the Max Planck Institute. We have also malaria, a small project, Frank Mockenhaupt, and we also have helminths, where is it? So warm infections, and that's a very good interaction also, and we won't hear today, other project, but good interactions with Susanne Hartmann with the Lepra Institute. And this is, and I think there will be more about this, the structure of this graduate school. This may look complicated, but Malia will allude on that also a little bit. There's a point system, how students can advance. It is all under the roof of the so-called Dahlem Research School, the FU, now also the Humboldt University received the status of the Excellence University. In Germany, only a few universities received that status, and by that, certain things were implemented to also help the students. There's other big projects like network grants, and so there's a big interaction once you are part of this research training group. Now to this acronym. And today, I have to admit, I'm a little bit proud on this part because this was really, I don't know, I think I included some other people, we did some brainstorming, but mainly there was one long night, and in the morning I told Lothar, wouldn't that make sense, this little acronym? So it was my idea, but it never made it into the official name. But it's the name behind it, and like we already said, it has a lot of double meanings. We can walk across the bridge. It binds to continents potentially, symbolically, of course. And what it stands for is Berlin, Hyderabad, Research on Infectious Diseases and Genetic Functional Epidemiology. So that means bridge, and it's still complicated, and I will explain a little bit what genetic epidemiology is. Actually, it's almost faded. Can you see this? It's supposed to be India. But the dot moved away. The dot is wrong, huh? We are more here, is that right? I think, and also Berlin, well, still works. Okay, and a little bit on history. We had the Vice Chancellor, he left already. And the, I think, predecessor of the Vice Chancellor was Professor Hasnain. And he's on this picture. And the first ideas for this collaboration were between him and Professor Hacker, who also is no longer in Berlin, but moved also upwards to a very prestigious organization in Germany. So the first talks, there was a memorandum signed, and then we collected abstracts as early as 2008, so 5 years ago, 22 projects. We had this first workshop, and the light is bad, but this year, and without Ty at this time, and it was a better idea. This is me, I think, talking to Thorsten Fischer, which is not visible. Anyway, then we went on. There was a delegation visiting again. We did a pre-proposal, that's how it goes with the DFG, also a long process, down to 15 projects. We had another workshop held here. And main proposal down to 13 projects, and finally surviving 12 projects, which actually 11 students funded. So, and then we started. We should, of course, extend this. Then the work started. Many visits. I don't want to list them all. We had a winter school here. We had, of course, seminars, joint seminars. I will show that in a little bit. And talking about Mitte, just briefly, and I used to show these pictures, and now that we moved away, I have no real reason to show them, but I will talk about TB a little bit, and so I put them back in again. This was where the Institute for Microbiology was until 1 year ago. We had to move out, and it has this nice lecture hall you can barely see. Some of you have visited, you may remember it. And also, around the corner is the campus of the old Charité, with somewhere here Angela Merkel, the Kanzleramt, and the new train station is somewhere here. Well, the light is too bad. It's in the center. And why am I mentioning this? Because this was the site where Robert Koch, 130 years ago, 1883, made the discovery of the mycobacterium, or he made the first announcement that those were the cause of TB. And in this region here, he had what they called barracks, where he was trying to treat patients. That never really worked with Robert Koch. But of course, the concept that bacteria cause disease, and that mycobacteria can be cultivated, and that he discovered so many bacteria, Pasteur in Paris also, of course. It was hallmarks for this whole field. I mean, microbiology started at that time. And until recently, we were in this building, and we may move close to this building again, but it's sold, so Charité is a little short on money, but historic place. So if you come, you will see a lot of history also. And I put it here one more time. This is really from the attic of our building, an old picture. This is Koch and many other people from his lab. And we have this room, until recently, we had this room with this green table, and it was the very table where he made this first announcement. Nia saw it. I didn't find the picture. I had one picture of you with a microscope. I couldn't find it. Anyway, this was the date. And by the way, the 24th of March is still worldwide the World Tuberculosis Day. Until today, you will hear all about the statistics on TB, and it's always the 24th of March where the news are full of these things. Okay, so a little bit of history. Now, Hyderabad. Of course, we also have a lot of institutions. They are across the city, and so there's some similarity. There's also some differences. If you want to go from here to here, in Berlin, you have a very good public transportation system. Here it's a little more adventurous. Whereas Abid, he once took me on a motorcycle to Blue Peter Institute. No helmet, of course. It was fun. It was great. And everything worked. So there are differences, yes. We had to learn this by sending students here doing work, but we got adjusted. I think differences also the other way when people come to cold Berlin and to maybe, I don't know, to all these forms that you have to fill until you can place an order. That's also complicated. So with cultural differences, we will hear about that, but it's very fruitful all in all, I would say, the interaction. Now, I promised I will explain genetic epidemiology a little bit. It's complicated, but it's the pathogen on one side, and microbiology was a driving force for genetics, for molecular biology for a long time. Small bacteria you could analyze, cloning was all done in bacteria. So bacteriology and genetics is very closely linked. Now, I think for maybe 10 years, the human genome has been solved, so now we can also look at the host, look at genetics. So we want pathogen and host genomics. We want to study the variation of host response to a pathogen. Immunology, but not everyone reacts the same way. One develops some fever and is fine, fights the bacterium. Others get chronic infections or very severe sepsis. So there's something to the genetics of the host. And that I think, Aeneas was also a key person in coining this new term, functional molecular epidemiology. And there are the projects, and again, ask me for the details. This is too much to go into all these, but they are all around this theme, and some of the, Katrin will talk, and Anne also. Is that right? Yeah. Yes, Mara. Where's Mara? There she is. So we will hear about it. Sorry, all these names. And more projects associated. And this was the definition. I think Nias put this into Wikipedia, and there we go. Now we have a definition. Functional molecular infection. So to make it short, epidemiology used to be that you only looked at germs, you typed them by genetics, and you said, okay, there's one class, there's the other class. That's it, taxonomy. We know, we name a bacterium, and we have a class, and they are related, and that's it. What we want to do now is to find out, is it related also to disease, to how it can spread in the host. And there, of course, different regions are also important. In different areas of the world, epidemiology is different. You may find the same taxonomic bacteria, but you may also find different ones according to the area and the hosts. So this is all more complex, but very fascinating. And that we want to emphasize on phenotypic and genotypic correlates, functionally relevant markers we want to find, and then we want to focus on these polymorphisms, and I will focus on that on my last part. One more time, pathogen and host. Variation of host, actually this is just a little bit differently worded. Functional molecular infection epidemiology is what we want to do. And yes, why interaction? We have expertise here in Hyderabad, bioinformatics, biotechnology, computational biology. We do protein modeling. We did that successfully already. We have then core facilities in Berlin. In my group, the SNP analysis, also bioinformatics in Berlin. Yeah, this can be exchanged, and this should facilitate also the students to succeed. These core elements, it's not only students getting some funds, but we also try to network and help them with the core facilities. This will be shown later also. We do different things also, summer, winter school, and so on. I think it will come again. Is that right? So I will not go into details. Of course, what then should be achieved, that the students, they climb up this stairway, they get a point system. There's also soft skill training, and also that will be mentioned later. And again, this summarizes expertise of Hyderabad and Berlin, and they are brought together. We also have animal models. I didn't mention that, at the Max Planck Institute in different places. Of course, molecular biology is one of the cores. But the last minutes, I wanted to talk about our project, which also has networking effects on other projects. It's also the project headed by Hortense Sleevogt that is relying on this cohort. And when I say we, it's the students, Nicole Dietrich, the postdoc Melanie Conrad, who have visited several times, Louis Berrocal of Hortense Sleevogt's group. They did a great job in organizing and setting up a patient and a control cohort. And it's also Abid sitting here who helped tremendously on this side to create this healthy volunteer cohort. So we talk about TB. And of course, there's much more TB here in India than in Germany. We cannot study it because there are no patients, but in the interaction, we can work on that. And we focused on a family of proteins called toll-like receptors with a strange TLR name. And they are important for recognizing bacteria. And we focus on this cluster, TLR2 with 1 and 6, because they recognize these lipopeptides, and they are present in mycobacteria. What we found as a surprise was there's another class of toll-like receptors within a cell, and they can sense RNA. And we are finding now that these receptors are also important for TB recognition.